Okay. Hello students, this is Kritika. So today we'll be starting with a new topic, forensic botany. So forensic botany is a branch of forensic science. So botany, we know that it is basically plant science. So how we are applying plant science to legal investigation to solve a lot of cases is what we are going to be dealing in forensic botany. So how does a plant act like a physical evidence to connect a suspect to the crime scene or a victim to the crime scene, or even to connect or link the suspect and the victim? So in order for that, how does a plant act like a physical evidence? Okay, so this can be used in a lot of things, like for identifying the location of the crime, for crime scene reconstruction. All of these things, forensic botany is highly useful for us. So we are going to first study about the nature of forensic botany. First of all, it is a multidisciplinary stream. That means it does not only involve botany, it also involves ecology, palynology. Palynology is basically the study of pollen grains. Then we have dendrology. Dendrology is basically about woody plants like trees, shrubs, vines. For these and all are coming under the study of all of these woody plants are coming up under dendrology. Then we have limnology. Limnology is basically about the aquatic systems, water ecosystem like lakes, oceans, ponds. All of these are getting covered in limnology. Okay. However, so you can see, right, the botany is covering not only botany, forensic botany is covering ecology, palynology, dendrology, limnology. Everything is linked with the forensic science. As I told you, plant evidence is highly abundant. It is transferable and is persistent. For example, seeds, pollen grains, and wood fragments and all, they get stuck to the clothing debris or to the vehicles or even to shoes and all, right? So they act as a very good physical evidence. And plants are immobile in nature and they are season specific as well. Season specific, so they are very, very informative. They tell in detail about the location of the scene, the time at which the crime was committed. Everything can be explained in detail. Okay, so that is about the forensic botany. Okay. Introduction. So we have the scope related to forensic botany as well. What are the scope means? First, it is helpful in crime scene reconstruction, as I told you. So identifying the plant debris on body or clothing, which will help in linking the suspect to the location and all, and estimating the time since death, that is post-mortem time interval. For example, if there is a plant growth or there is decomposition stages of plants and all, that will be helpful in indicating or predicting the time since death, the post-mortem interval. PMI stands for post-mortem interval. Post-mortem interval. See, there are a lot of seasonal plants. Seasonal plants basically they act like silent witnesses. So the another name we can call is silent witnesses. Silent witnesses, because they play a very important role at indicating, okay, this particular time the crime was committed. Okay, so for example, there are certain annual plants are there, deciduous plants are all there, which all is indicating. For example, in Europe is one of the plant is called as willow herb. Willow herb, this is basically like in European region for forensic cases and all to solve it. This particular plant is considered like the holy grail. Okay, especially in terms of like what to say, in autumn season and all. No, this particular plant is formed. It's then they know that willow herb mainly grows in autumn season. So the crime was committed in autumn season. For indication of all these values, even maple, oak tree, all these act as a good indicators, because maple and oak tree and the decomposing stages of the body and all, these are some of the plants which is seen in the decomposing bodies and all. Under the remains of the body, these are beautifully decomposed. So again, they act as a very good physical evidences in decomposing remains of the body and all. Okay. Hmm. Next is in scope of the again scope we did Scope we did till the Caesar plant evidence. Then they are used in drug-related cases as well. So an identification of cannabis plant or Papaver somniferum, that is you get opium and poppy from it, and erythroxylum coca, which is basically you get cocaine and all. And wood identification cases, especially in smuggling of timber, sandalwood, in case of weapon or furniture and all, wood identification. And then we have diatom analysis. So diatoms are basically a kind of specimen that is found in water bodies and all, and they are extremely resistant to high temperature, very high temperature or very cool temperature, any kind of atmospheric or climatic condition they can withstand it. That is why diatoms are used in soundproofing of the walls and everything. So diatoms are mainly used in drowning cases because diatoms goes and accumulates in the femur bone. We know, right? Femur is a thigh bone, longest bone of your body. So all of the diatoms goes and gets accumulated in your femur bone. So for drowning cases and all, diatoms are very good, source of evidence as well. Then we have trace evidence like the pollen grains or spores on clothing and all. And the study of pollen grains is called as palynology. So the pollen or spores in clothing, which will also help in connecting or linking the vehicles and or suspect to the crime scene and all. Then it is used in wildlife cases, illegal trading, that is poaching of animals, especially protected plant species are all there. So if you're doing illegal trading of even plant as well as animal species, so that can also be used in identification. Then we have in agricultural environmental crimes, that is in poisoning cases. There are a lot of plant poisons as well. For example, strychnine is there, a lot of plant poisons are there. Okay, Datura. So those are all coming under agricultural environmental crimes as well. Okay. Now coming to wood and their identification. So wood are used in weapons and all. For example, to make lattis. Latti is used in police people and all. That clubs, furniture for packing materials and all. Or in cases of illegal logging of a timber smuggling and all, wood is basically used. So matching of the wood fragment that you obtained from the crime scene to the source or the sample, if you match it together, we can establish the important link between the crime scene as well as the wood fragment and all. Okay, for example, let us take the structure of wood relevant identification. There is hardwood and sapwood. There are two type of woods, right? They both are different in color, the density, everything is totally different here. And about the annual rings. So in the tree, if you see, means that growth rings will be there. That it basically determines the age of the tree. So these annual rings or age rings are formed in the tree. It is due to the seasonal growth only. Okay. So this again is used for dating of the trees. That is basically predicting the age of the tree, and for species identification and all, the annual rings or growth rings in the trees are very helpful. Now coming to vessels. So we know xylem elements means in schools we have studied, right? We have plants basically have two type of tissues, vascular tissue or vascular bundle. So basically the two vessels are nothing but xylem elements. So we have xylem and phloem will be there. So xylem is a tissue which is present in plant which will helps in conduction of water and nutrients from the root to the plant parts. And phloem is another tissue that is present in the plant which helps in the conduction of the food to all the parts of the plant. So now here in this case we are talking about the xylem vessels. So the arrangement of xylem vessels also is very species specific. That is also a very good source of information. Then we have parenchymal cells. So we know plants are made up of different kind of cells like parenchymal cells, collenchyma cells, sclerenchyma cells. So a lot of cells are there. So this parenchyma is basically like a food storage tissues or cells that is present in the parenchymal cells basically forms the parenchymal tissues. They are basically the food storing tissues. Okay. So species to species, the parenchymal cells will differ. The shape, the size, the storage of food, everything will basically differ. Okay. So for some cases they are used in food storage. For some cases they are not. They are inactive in nature. So plant specific also. So when we get the parenchymal cells, so depending upon the arrangement, the width, the spacing, and all, it will vary from species to species. Then we have tracheids and fibers. Again, these are also additional elements that is present in your plant. For example, this is present in your soft woods, that is in gymnosperms and all, versus in hardwoods in angiosperms. So tracheids will be present mainly in gymnosperms and fibers will be mainly present in your angiosperms. Then we have resin canals that is present in your coniferous plants like pine trees and all. No, resin canals be present here. As you can see, right, each and every plant has unique type of features, tissues, different type of elements is present there. Now, Now you can see wood and their identification. Identification of wood means see, you have teak wood, you have oak, pine, mahogany, rosewood. So different type of wood. They are of different colors, different textures, different grain pattern, weight, hardness. Everything will get basically they are differing. Then there are different methods of identification. First is microscopic examination. You see the color, you see the grain pattern, you see the surface, the texture, the odor, the density of the wood, the weight of the wood. Everything you are doing is macroscopic examination. Sorry, I said microscopic. Macroscopic examination. Microscopic means we are taking a small section of the wood fragment and we are observing it under the microscope. We are seeing the radial section, the transverse section, the tangential section, everything, section by section. The proper cellular arrangement of the cells in the wood fragment can be observed under a microscope clearly. Okay, so then we are doing reference with the comparison with the reference would also that is obtained or in databases will be there. We can compare it and see. Okay, so reference would and the question would we both they will take and we can compare it and see. Then we can do some chemical testing. For example, for resins, tannins, and for lignin content and all, we can do the chemical testing as well. So, for DNA analysis of wood, for species level and for individual tree matching level, in case of like timber smuggling cases, individual tree matching level can be. So, which particular tree did they actually took the timber smuggling cases was used and all. So, for all of these things and all, DNA analysis of wood for species level, for individual tree matching level and all can be utilized. Okay. So you can see here anatomical features like vessel diameter, as I told you, width, the presence of xylem, phloem, tracheids, fibers, or parenchymal tissues, how is the arrangement of the tissues. All these are certain anatomical features which will differ from plant to plant. So when you're doing the matching of a plant, basically, especially in terms of wood and all, what you're doing is that you're comparing the cut surface or the feature pattern of the wood to the wound on the victim. For example, the victim has got injured through because of a wood piece. So we are seeing that the cut surface or fracture pattern of the wood to the wound on the pattern or on the victim. That is tool mark analysis to do it and all. No, we can basically match it and see splinters. I told you, small cuts and all which is recovered from the crime scene or vehicle which suspected object, any broken furniture or anything that is involved there, or you're matching the growth rings and all. All of these things are very good evidences. So all tool mark analysis, any kind of wound or injury in case of victims and all for vehicle, especially in what to say, vehicular accidents and all. No, so those cases, a log or the plank of wood or the wood that is obtained from the crime scene from the suspect's premises and all is very much important evidence. So as you can see, matching importance. So matching is basically a process of comparing the evidence that is obtained from the crime scene to with a known sample to establish the link. As you can see here, it helps in connecting the suspect to the crime scene. It is a very strong support in investigations and all. Okay, it gives us accurate and reliable analysis as well. So now coming to diatoms. So this is a microscopic examination of diatoms. So it is basically a unicellular algae only. And the cell wall is made up of silica. That is why they are very, very resistant and strong in nature. They are highly found in water bodies like ponds, rivers, oceans, everything, seas, everything. So why in terms of forensic science diatoms are very important is that they are very hard. They have resistant silica shell. So they can survive any kind of condition, decomposition, putrefaction, and even cremation also they can survive. That strong they are. And one more important quality of diatoms is that each and every water body, the diatoms will differ. So they have the ability to change it according to the climate, according to the water body. They have the ability to change them. So they can survive in any kind of water. So each and every water body, you can find different type of diatoms. Okay, so this particular pond is having a characteristic diatom flora. This particular sea is having some other diatom flora. So it is very unique or peculiar in nature. So if they're in case of drowning cases and all means, so this particular diatom can be found in this ocean only or this sea only. So this person might have been drowned in this particular, because what happens is that the person would have been killed and the body would have been, like for example, they would have been drowned in one particular ocean and the body would be somewhere else. Okay, so it will be at the secondary crime scene basically. Okay, you have primary crime scene is the main area of crime that is committed. Then the body might be recovered in a secondary crime scene. So in order to mark like which particular sea or water body was the person drowned, so to find out that diatoms are very useful. As I told you, diatoms are specific in each and every water body. So by looking at the femur post-mortem analysis, in femur only all the diatoms will be accumulated. Okay, so in femur, we know that longest thigh bone in our body, there only all the diatoms will go and accumulate. When they are doing post-mortem analysis, they can take the diatom and the diatom that is obtained from the water body. They can take the reference and question sample. They can do analysis, comparative analysis, and they can find out which particular water body was the person drowned. Whether it is a suicidal death or an accidental death, diatoms are extremely helpful. Okay, so as I told you, diatom test and drowning and all. So if the person is alive when entering the water, the water containing diatoms will directly go into the lungs of the person. So diatoms will pass through the alveolar capillary membrane, then it goes to the bloodstream, then to your organs, and then to your bone marrow. That is your femur, or to the sternum it goes. Okay, then to your brain, kidney, liver. So all your internal organs are completely filled with diatoms now. So presence of diatoms in the distant organs, especially in case of bone marrow, is a very strong evidence to show that the person has antemortem drowning. That means the person, say post-mortem drowning means the person has been killed and the body was dumped into the water body. Antemortem drowning means a person was alive when he was drowned inside the water body. Okay, the person was alive when he was submerged inside the water body. So if the body was thrown into the water after death, that is post-mortem, after it's done, after the person has dead, then the water body is thrown into the water means. So then diatoms might have been stored only in your lungs and stomach. They might not go into the bone marrow and all, because obviously if the person is dead means the blood circulation will be stopped. And bone marrow is where the production of RBCs basically takes place. So the person is dead means no circulation will take place. So you can't find out diatoms in the bone marrow and all. It will be stopped only to lungs and stomach. So based on this, you can identify if the person or the body is undergone post-mortem drowning or ante-mortem drowning respectively. Clear? So now what is the procedure to do diatom analysis? This is again a very important topic. As I told you, tissue samples either from femur or from bone marrow or from lung or liver, any internal organs, you can take the diatoms are collected and all of the organs are digested using some concentrated acids like HCl or nitric acid or sulfuric acid. We basically digest it to destroy all the organic matter. Only the hard silica frustules, the outermost covering of the diatoms alone will be present there. This residue can be examined under the microscope to find out how many number of diatoms are there, what is a particular species of diatoms that is obtained. Everything can be observed in detail. That is your diatom testing. Clear? Now, the next is about the forensic significance. As I told you, for confirming the drowning cases of cause of death, differentiating antemortem versus postmortem submersion, diatom species or type analysis. What type of diatom or what particular species of diatoms is especially useful in establishing the location of the crime. And in advanced decomposition cases or skeletal remains are only found in the crime scene means by examining if there is silical frustules that is obtained means okay that person is, that means the diatom is persistent. So even in advanced skeletal decomposition cases also diatoms are extremely helpful. Okay, so now till I hope till diatoms is clear. Now we are going to start with forensic palynology. That is a how pollen grains are helpful in solving forensic cases. See, pollen grains are nothing but the male gametophyte of flowering plants will basically produce pollen grains. So this pollen grains, if you see the structure of pollen grains means, so in school we have studied like exine, intine. So basically the structure of pollen grain has an extremely durable, very strong outer mold called as exine that is made up of something called as sporopollen. This is one of the most as well chemically resistant biological substance. This sporopollen is very resistant. Heat, cold, rain, acid treatment, any kind of climatic condition, they can survive and thrive for thousands of years. So sporopollen has that type of ability. So you can see that pollen how strong they are in case of forensic evidence and all. So how these pollens are forensically useful means you can get for pollen grains in enormous quantities. They are very small, they're microscopic in nature, but you can get it in enormous quantities. And they easily get stuck to the surface of skin, hair, vehicles, dress, tools, everywhere. So they are basically pollen assemblages. So different type of pollen grains basically assembled together. And as similar to diatoms, they are also highly specific to their geographic location. Season-wise plant and all, different plants at different times and places and all, you can get it. So that is why pollen grains are extremely useful. One of the reasons. Mhm. See, pollen grains are microscopic in nature. They are produced by the anther of the flowers, basically. So they help in plant reproduction. And when pollen grains go to the stigma of the plant and then stigma style ovary, then fertilization reaction takes place. So basically in flowering plants in school, we would have studied this chapter, right? That same particular thing only. So it is mainly used in forensic science, in archaeology, in pollen analysis and all to understand about the past climatic condition, the past ecosystem and all, pollen grains are highly helpful for us. So now coming to the identification and features of pollen grains, as I told you, enormous quantity of pollen grains can be found and the size and shape they are very different. So you can get oval, spherical, triangular, different shape and sizes you can obtain. And then we have the aperture type. Okay, for example, pores or furrows and all, monoporate, triporate, again that and all is there. And surface ornamentation means sculpturing. Exine I told you, the outermost covering is different plants. They have different type of pollen grains will have different type of structures. Some will have like smooth, some will have piney exine, some will have reticulate, some will have striate pattern. And different type of the outer structure of exine will be different for different plants and all. And symmetry also, whether it's a radial symmetry or a bilateral symmetry, that again, so it's very unique for different type of pollen grains. So what is the method of study here? You're collecting the sample either from the dress or skin or hair, body, vehicle, soil, anywhere you can collect it from the crime scene. Then we are doing acetolysis. Acetolysis means basically it is a chemical treatment where we remove all the organic content and we isolate only the pollen exine and we'll be examining the pollen exine in detail. What is a particular species of pollen and what type of exine do they have? Do they have smooth or straight or any kind of pattern, spiny pattern? We'll basically examine them. So first we will examine under the light microscope or through scanning electron microscope, that is SEM, for detailed surface analysis of the exine. Then we will compare this particular pollen grain with a reference or database pollen grains of that particular region and we'll see whether this particular pollen grain matches with that region or not. So we can identify that particular pollen grain has matched this region. Okay, this region only the person, the crime must have been committed. Like that, we can identify it. So location of crime scene to identify this pollen grains are also very helpful, similar to diatoms. And coming to applications. So I told you linkage of a suspect to a specific crime scene, determining the season of death or crime based upon the pollen, because there'll be certain seasonally based plants also, depending upon the seasonally based plant pollen you can identify. Then tracking the geographic region, okay, and establishing whether the body has been moved from its original location or not to identify it. All of these things, diatoms are extremely, sorry, pollen grains are extremely helpful. So now coming to your DNA, that is DNA is basically deoxyribonucleic acid. We know, right? It's a genetic material present in all the living organisms and all. And we know the structure of DNA. So it is a double standard helical structure. It is mainly made up of a sugar called as deoxyribose sugar. So the backbone of the DNA is made up of deoxyribose sugar, phosphate group, and nitrogenous bases are there. So mainly we have four nitrogenous bases in DNA. That is basically adenine, guanine, and thymine and cytosine is there. So that is basically adenine and guanine comes under purine, cytosine and thymine comes under pyrimidine. So these are the four nitrogenous bases that is present in your DNA. And we know how the pairing happens also. Adenine always pairs with thymine and guanine always pairs up with the cytosine. And we know this, right? Adenine always pairs with thymine. There are two hydrogen bonds present between adenine and thymine. And guanine always pairs up with the cytosine, means there is three hydrogen bonds between them. So we can know that guanine to cytosine, this bond to break, we need lot of energy than compared to the bond between adenine and thymine. So there will be hydrogen bonds that is present between both, all the four nitrogenous bases. Clear? Now, so the double helical model. So we know double helical model is there. Two strands are basically running past each other. Okay, so this double helical model of DNA was proposed by Watson and Crick in 1953. They told that two anti-parallel polynucleotide chains are coiled around each other, and the backbone of the DNA is made up of sugar, phosphate, and all. And we have four nitrogenous bases, that is purines and pyrimidids. And I told you adenine to thymine, there are two hydrogen bonds. Cytosine and guanine have three hydrogen bonds. So this is a complementary base pairing that should happen. Always adenine pairs up with thymine and always guanine pairs up with the cytosine. That is the complementary base pairing. And one complete turn of an helix is basically, there is approximately 10 base pairs, right? For one approximate or complete turn of the helix is about 3.4 nanometer. Diameter is about 2 nanometers there. So there are major groups also there and there are minor groups as well. This will help in allowing the protein binding to take place properly. Okay. So now coming to the different forms of DNA. We have bDNA, genomic DNA is there, and we also have polymorphism in DNA.